Bismillah ar-Rahman ar-Rahim. I will continue discussing the histology of the eye. We finished in the previous lecture the eyeball, the three coats of the eyeball. We discussed the cornea, the sclera, which are the fibrous, outer fibrous coat. We discussed the middle coat, the uveal tract, including the iris, ciliary body, and choroid. And now we have to discuss the histology of the lens and go to the inner coat of the eye. First, let's revise what's refractive media of the eye. These are transparent media that permit the passage of light to the retina, which include the cornea first. This is a transparent structure, the aqueous humor present in the anterior posterior chamber, and the third one is, this is our structure, the crystalline lens, okay? And then the vitreous, which is a transparent refractile gel-like structure present in this large cavity in the eye and forming the vitreous, which is behind the lens. This is the vitreous body. What about the crystalline lens? What's the histological structure of the crystalline lens? From its name, it's transparent. So it's a transparent biconvex avascular structure. Again, remember, the cornea was avascular, no blood vessels, it was transparent. Also here in the lens, it is transparent, there is no blood vessels in it, so it's a transparent biconvex, as we can see, avascular structure. And also another property is the high elastic property. So it can change its curvature from being globular to being stretched, flattened. This is a very important character for accommodation, for near vision and for far vision. It's situated immediately behind the pupil. You can see here the pupil opening in the iris here, and it's situated just behind the pupil. This is the lens. Again, this is the cornea, anterior chamber. This is the iris, pupil, the crystalline lens. As you can see, it will change its shape during accommodation to near and far vision from the flattened to more globular shape. What's the histological structure? You'll find that this is the lens, okay? It's formed of three layers. From outside, it's covered with capsule, which is a connective tissue or basement membrane material, okay? And this capsule will give attachment to what? To zonular fibers. Zonular fibers, you remember the ciliary body, ciliary processes, and these ciliary processes will give the zonular fibers, or we call it suspensory ligament of the lens, okay? This will keep the lens in position. So the capsule gives attachment to these zonular fibers. Underneath the capsule, we'll find subcapsular epithelium, a layer of epithelium which is present only on the anterior surface of the iris. And most important is the lens fibers. So the lens is formed of closely packed, elongated, parallel to each other lens fibers. So the most important for you is the capsule, which give attachment to the zonular fibers, and the lens fibers, which are the transparent structure, elongated, differentiated cells. We call them lens fibers. They are closely packed, okay? They are very transparent because they contain a protein called crystallin, okay? It's very transparent. This is histological section stained with H and E to show you capsule from outside, subcapsular epithelium underneath, and this we are interested here in these lens fibers, unique cells losing their nuclei. There is no nuclei in these lens fibers. They are closely packed, parallel to each other, as you can see, and very crystalline, very transparent. No blood vessels are present, no nuclei are present, closely packed together, very regular, and containing a protein called crestin. This will contribute to its transparency. Again, this is the shape of the capsule, epithelium, and then the lens fibers. So the lens is transparent, okay? Crystalline lens is very transparent, but once opacification occurs in this lens, it will lead to cataract, which is a clinical condition. Also, the lens is responsible for accommodation for near and far vision, because as you can see this by the zonular fibers, when relaxation or contraction or traction or tension on these zonular fibers together with the action of the ciliary muscle will change the lens and will be adapted for the accommodation. By tension on the lens by these zonular fibers, this will change the curvature of the lens and help for accommodation for near and far vision. Then we'll go to the most important inner coat of the eye, which is the retina, the photosensitive retina. What's the function of the retina? You know that it's responsible for the accurate analysis of the form, light intensity, and color reflected from objects, and also it encodes these parameters into electrical impulses for transmission to the brain. The retina is composed of two main parts. An outer part, this outer part is the pigmented part. This is the epithelial, retinal pigmented epithelial layer. This is the non-photosensitive retina. And then the rest of the layers, these are the retina proper, the sensitive retina, and these are responsible for the sensory retina. So we have two main parts, the outer part, the pigmented epithelial cells, retinal pigmented epithelial cells, and then the retina proper, which is the sensitive part of the retina. The retina is formed of 10 parallel layers from outside inward. From outside inward, as I said, the first is the pigmented retinal epithelial cells, which is underneath is the choroid. So this is the outer part, while the rest of the layer, layer number 10, will be the most innermost layer, and this is facing what? The vitreous inside the eye. So from outer to inner, the retina is formed of 10 layers. What are these 10 layers? These 10 layers will include, first, pigment epithelium, this layer of retinal pigment epithelium or pigmented epithelium. Then you will see multiple layers of retina proper, starting with rods and cones. These are the rods and cones. The photoreceptors. These are the photoreceptors. Then number 3 is a line. We call it the outer limiting membrane. And then you will find the nuclei and cell bodies of rods and cones in a layer called outer nuclear layer. You can see in histological section, this is diagram to show you the cells, and the histological section will show you the most prominent structure. Here we can see the nuclei, aggregation of nuclei. This will denote the cell bodies and nuclei of the photoreceptors. These photoreceptors, which are rods and cone, have an outer segment will act as a dendrite, and here it has a synaptic terminal which will act as an axon. So this is the dendrite of the rods and cones, and this is the axon terminal of rods and cones. When axon terminal synapse with dendrites of the layer above, they form a layer faintly stained in the form of fibers here in histological section called plexiform layer. So what's the plexiform layer? This is a layer of fiber, synaptic contact between axon terminal and dendrites of cells, forming this layer. So the axon terminals of rods and cones, together with the dendrites of the layer above, which is mainly the bipolar cells. So now we know that there are rods and cones, photoreceptors in blue, 2 types of cells, and then there is conducting neurons, which are, one is bipolar, this bipolar cell spindle shaped, as you can see, cell body, and one process will act as a dendrite, the other process will act as axon. These bipolar cells, together with other cells I mentioned, will synapse in this layer, which is called outer plexiform layer, this faint layer. Then we'll go to layer number 6, which is the inner nuclear layer. Notice that we have an outer nuclear layer, this one, formed by cell bodies and nuclei of rods and cone. Then we'll go inner nuclear layer, and we'll mention what are the cells sharing in this, our nuclei of cells sharing in this layer. And in between, there is outer plexiform layer. What about this inner nuclear layer? There are 4 cells with their nuclei and cell bodies forming this inner nuclear layer. These cells, mainly bipolar cells, these red cells, bipolar cells. Then we have 2, we call it associating cells. This will help in the synaptic contacts, in propagation of the stimulus. So we have the horizontal cell and the amacrine cell. So what's present in the inner nucleus is cell bodies and nuclei of bipolar together with 2 cells, horizontal cell and amacrine cell. So these are 3 cells. The 4th cell is called Muller cell. What is this Muller cell? You see this one extending from the innermost layer to outer limiting membrane. This cell is called Muller cell. What are these Muller cells? These are supporting cells. So they are not neurons. They are not conducting impulse, but they are supporting cells, nutritive cells. Okay, it's a neuroglial cell. You remember in the brain you have a nerve cells and supporting cells or neuroglial cell. This is just like in the brain. It is neuroglial cell. They are supporting cells, nutritive cells, which extends from the inner limiting membrane to the outer limiting membrane, which is layer number 3. Okay, its cell body and nuclei are present in this layer, which is the inner nuclear layer. So the inner nuclear layer, this encloses the nuclei cell bodies of 4 cells: bipolar, horizontal and amacrine cells, together with the supporting cells, which is the Muller cells. Then we'll go to layer number 7. It is the inner plexiform layer. Again, what's the inner plexiform? Plexiform means synaptic contact between processes. Here, axon terminals of bipolar together with dendrites of a neuron above, very important, called ganglion cells. In this, there is synaptic interplay, or there is a synaptic contact here, comprises the layer of inner plexiform layer. Again, it's fiber, so it appears faint in the histological section. Then we'll go to layer number 8. It's formed of ganglion cell layers. These are the ganglion cell layers. Ganglion cell layers, large cells with large vesicular nucleus, and this is the layer of the ganglion cell layers with its nuclei. Then we said that the dendrites of this ganglion cell layer will synapse in the inner plexiform. So what about the axons of these cells? The axons of this ganglion cell will form the optic nerve fibers, the nerve fibers layer. Okay, and here again it's a faint layer in histological section because it's formed of fibers. So the axons of the ganglion cells will form the layer number 9, which is the nerve fibers. It will go like this, perpendicular, and then will go outside at optic nerve fibers. Then the innermost layer is the inner limiting membrane facing the vitreous body. So these are 10 layers. We'll repeat the layers again until you can correlate the histological diagram and cells with the histological section of the retina, and we'll discuss one by one the 10 layers of the retina. As we said, what are the neurons or the cells present in the retina? There is photoreceptors. These are modified neurons, which are rods and cones, the blue cells. These are rods and cones. Now we'll discriminate between the rods and cones in shape. Then we have conducting neurons, will conduct the impulse. These are the bipolar mainly and the ganglion cells. We have also association neuron like horizontal and amacrine cell. And another category, which is the neuroglial cell, supporting cells. This example here is the Muller cell, which is span the whole thickness, or most of the length of the retina, extending from the inner limiting membrane to the outer limiting membrane, and the cell body is present in the inner nuclear layer. Let's start the layers one by one. First is retinal pigmented epithelium. Again, I said this is the outermost, it's not sensitive. Okay? Resting on what? What's below? It's the middle layer, which is the choroid, containing blood vessels here and pigments. So the first layer, outer layer, is the retinal pigmented epithelium. From its name, pigmented, it contains melanin, and the function will be absorption of extra light. Then, number two, this is part of rods and cones. Which part? Mainly the outer and part of the inner segments, or the inner segments of the rods and cones. We'll discuss now the histology, but you have to know that layer number 2 is the layers of rods and cones. Rods and cones with their outer segment, inner segment, not the cell body, because the cell body and the nucleus is present in layer number 4. We'll go to the layer number 3, outer limiting membrane. What's this outer limiting membrane? I said that the molar cells or the supporting cells extends from the outer limiting membrane to the inner limiting membrane. What will happen? Why this appears as a limiting membrane? Because they said that this membrane is seen by electron microscope due to junctional complexes between the molar cell and the rods and cones. So the junctional complex between the molar cells and rods and cones forming a line, dark line EM. So we identify this line as a layer called outer limiting membrane. Actually, what's the outer limiting membrane? It's junctional complex between the molar cells and the rods and cones. Then we'll go to layer number 4. In layer number 4, we can find the outer nuclear layer. What's the outer nuclear layer? As I said, it encloses the cell bodies of the rods and cones containing the nucleus. This is the cone cells in pink, and these are the rod cells in blue. The nuclei and cell body of these rods and cones are present in this layer, which is outer nuclear layer. You can find that the nuclei of the cones are larger, fainter, present at one line opposite the outer at the level of the outer limiting membrane, while the nuclei of the rods are at different levels, much smaller and darker. Then layer number 5, outer plexiform layer. What's plexiform layer? Synaptic contact between fibers, including here, as I said, the outer segment will act as a dendrite, while towards the synaptic terminal here, this is the synaptic terminal of rods and cones. Synaptic terminals of rods and cones will synapse with the dendrites of the layer above, mainly cells of bipolar together with the horizontal cell. So the outer plexiform display the synaptic connections between axon terminals of rods and cones and dendrites of bipolar cells and horizontal cell. Again, notice outer nuclear, inner nuclear, in between outer plexiform and then inner plexiform. So rods and retinal pigmented epithelium from outside, rods and cones, then outer limiting membrane, outer nuclear, outer plexiform, then we'll go to inner nuclear. Inner nuclear, remember, there are cell bodies and nuclei of 4 cells. What are these 4 cells? Mainly the bipolar cells. And the dendrites of bipolar cell will synapse in which layer? In the outer plexiform, while its axon will synapse where? Inner plexiform. So these cells will discuss it as extending from the outer plexiform to the inner plexiform layer. Now, what are the 4 cells present in the inner nuclear layer? 4 cells present in the inner nuclear layer. Number 1 is bipolar cells, cell body and nucleus of bipolar cells. 2 associating cells, you remember, one is horizontal cell, the other is amacrine cell. What about the 4th cell? 4th cell is the molar cells, the molar cell or the supporting cells, the neuroglial cells. Again, outer plexiform, inner nuclear, inner plexiform. Here, what's left from this bipolar cell? It's shared with its dendrites, their dendrites share in synaptic contact in the outer plexiform. What about their axon terminal? Axon terminal together with the dendrites of the ganglion cell layer will occur here. This synaptic contact in the inner plexiform is between axon terminal of bipolar cells and the dendrites of ganglion cells together with the amacrine cells. Synapses with the amacrine cells. Then we'll go to the layer number 8, which is the ganglion cell layer. This ganglion cell layer, large cells with large vesicular nucleus present in this layer. Their dendrite synapse with the axons of the bipolar cells and amacrine in the inner plexiform layer, while their dendrites share here, while their axons will bend, forming the optic nerve fibers. So layer number 9 is the layer of optic nerve fibers. You see, they are piercing the sclera at lamina cribrosa and go out as optic nerve fiber, and it becomes myelinated by oligodendrocytes. This the formation of the optic nerve fibers. And the innermost layer will be the inner limiting membrane. What is this inner limiting membrane? Formed of what? Remember, outer limiting membrane is the tight junction between molar cells and rods and cones, while inner limiting membrane, this is the basal lamina of the molar cell. So the molar cell extending from inner limiting membrane to the outer limiting membrane, here by tight junction and here by their basal lamina. See, notice that the light goes from here, from the inside of the eye through the cornea, anterior posterior chamber, lens, vitreous, and will go here, while the stimulation, this is the pathway of light, enters from inner to outer, while you can see that stimulus propagation of impulses will go in the opposite direction till it go outside through the optic nerve fibers. Let's start layer by layer. Retinal pigmented epithelial. Retinal pigmented epithelial, as I said, this is the outer layer. It's not sensitive. It's not photosensitive layer. This layer are cuboidal cell, as you can see here. They are cuboidal cell with rounded nucleus, but there is basal foldation of its membrane and apical microvilli. So it has these two characters, basal foldation, and we know that basal foldation means important for adaptation for ion transport. So there is here exchange and ion transport through this the foldation in the basal layer. We know that beneath it, outer to it, is the choroid containing the blood vessels, while the apical part of the retinal pigmented epithelium contains multiple microvilli, and here they are in contact to the outer segments of rods and cones. What are the functions of the retinal pigmented epithelium? There are main 4 functions for the retinal pigmented epithelium. First of all is absorption of light. Absorption of light after stimulation of the photoreceptors, you can see that the extra light is absorbed by the melanin present in these cells. So that's why they are pigmented, for absorption of extra light. This is function number 1. Function number 2. We can notice that in between these pigmented epithelial cells or retinal pigmented epithelium, tight junctions between the cell. When we see tight junctions between the cell, this means there is barrier. Barrier between what? This is the photosensitive layer. The rest of the layers is the delicate photosensitive layers or part of the retina. There must be barrier between the blood vessels present here in the choroid and between the photosensitive retina. This is through tight junctions between the cells to keep the toxic, blood-borne toxic material to go inside the photosensitive retina. So it's protected from blood-borne toxic material through this barrier, which is tight junction between the retinal pigmented epithelial cells. Also, there is another part of the retinal barrier. This is part of blood retinal barrier. Remember, we have blood aqueous barrier, and now we have blood retinal barrier. and this blood-retinal barrier is formed of two components. One of them is the tight junctions between the retinal pigmented epithelial cells. The second one is tight junctions between the endothelial cells of the capillaries, which is present mainly in the ganglion cell layers or among the layers of the retina. There is some blood vessels where the endothelial cells of this capillaries are held together with tight junctions. So the tight junctions between the capillary endothelium and between the retinal pigmented epithelial cells will constitute or form the blood retinal barrier to protect the retina. Number 3, we have restoration of the photosensitivity. You know that we have visual pigments present in the rods and cones, and part of it to restore its photosensitivity, vitamin A is required. Here, esterification of vitamin A is through the smooth endoplasmic reticulum present in this retinal pigmented epithelial cell. So the modification of these cells to adapt its function is, number 1, presence of melanin for absorption of extra light, the junctions, complex tight junctions between the cells for formation blood-brain barrier. Number 3, presence of numerous smooth endoplasmic reticulum, which helps in esterification of vitamin A, and so restoration of the photosensitivity. The last function is, as I said, the apical part of the cell contains microvilli, which is related to the outer segments of rods and cones, and so it helps in phagocytosing the tips of this outer segments for renewal of the photoreceptors. So which cell is responsible for renewal of the photoreceptors, rods and cones? It is the pigmented epithelial cell, or retinal pigmented epithelial cells, by phagocytosing the tips. Which organelle will be responsible for phagocytosing this tips is presence of lysosome. So it has melanin pigment for extra light absorption. It has the lysosomes for phagocytosing, smooth endoplasmic reticulum for esterification of vitamin A, and tight junctions between the cells for formation of blood-retinal barrier. Then we go to layer number 2. Again, this is the layer of rods and cones. As I said, rods and cones contains the outer segments of rods and cones and the inner segments of the rods and cones. Rods and cones are present, as you can see, they are parallel to each other, okay, and they are oriented perpendicular to the retina, okay. They are parallel to each other and perpendicular to the retina, and they are light is induced or stimulation occurs by hyperpolarization. If we look at the rod and cone, this is a rod cell and we have a cone cell. What we are interested in is the presence of what's called outer segment and inner segments. In the outer segments, we can see that visual pigment, which is called rhodopsin. Rod cell contains what? Rhodopsin. Visual pigments are present in this outer segment, while the inner segment is the metabolic part of the inner segment, which contains the organelles like mitochondria, rough endoplasmic reticulum, Golgi apparatus. So synthesis occurs in the inner pigment, and we have part of the rod cell containing the nucleus. And remember that the nuclei of rods and cones are present in a layer called outer nuclear layer, and the outer segments will act as an dendrite, while there is synaptic terminal. This is the synaptic region. In case of rod, is called spherule. Again, this is a diagram to show you the outer segment and inner segment. The outer segment is formed of what? It's formed of membrane, disc-shaped enclosed membrane, okay, or disc-shaped we can see the rod cell, and the rod cell is formed from the outer segment, which will act as a dendrite, and we have here the inner segment, metabolic part, which contains the organelles, the cell body and the nucleus, and the synaptic terminal. What's the outer segment? The outer segment is a modified dendrite. It's formed of numerous flattened membranous discs. It's formed of membranous what? Membranous discs. Discs is formed by cell membrane, and these discs will enclose the visual purple or visual pigment, which is called rhodopsin. So by EM we can see now the outer segment in form of these discs. Higher magnification to show you the discs. And the metabolic part is the inner segments containing organelles, mitochondria, rough endoplasmic reticulum, Golgi, and polysome. Here the nuclear region containing the cell body, and by the end the synaptic terminal. If we look at this histological section, you can see this is the choroid, and this is the pigmented epithelial cells. And now we can see here this part is the outer and inner segments of the rods and cone, or layer number 2, which is the layer of rods and cones. This is a comparison between the rod cell and the cone cell. What's the difference? We're interested here in the outer segment. Look at the outer segment. If it is tall and cylindrical, it is the outer segment of the rod cell. If it is short and triangular, it is the outer segment of the cone cell. Okay? This is the synaptic terminal. This is called spheroid. This is pedicle. Yeah, it's not important to know the names, but the most important is to know the shape of the outer segment, which will act as a dendrite containing the discs, okay, which will contain the visual pigments. Rhodopsin in case of rod and iodopsin in case of cone. Inner segment, as I said, containing metabolic part, while nuclei or cell bodies, okay, as I said, the nucleus of the cone is large and faint in color, present at the line of outer limiting membrane. Remember, what's the outer limiting membrane? I said, you see this rod cell is surrounded by a neuroglial cell called Muller cell, supporting cell. Also the cone is surrounded by supporting cells. Junctional complexes between these supporting cells, Muller cells, and the rods and cones forming a line in the EM called outer limiting membrane. So now we understand the outer layer, outer retinal pigmented epithelial layer, then the rods and cones, we mean outer or inner segments of the rods and cones, then layer number 3, outer limiting membrane, then layer number 4, which contains the cell body and nuclei of rods and cones. Again, these are the rods and cones, cone with triangular outer, the outer segment, and we have here the nuclei. Difference in rods and cones, many difference, much more number the rod cell, while about 6 million the cone cells. And we have to know that it's responsible, rod cell responsible for night vision, while the cone cell is responsible for the acuity of vision and the color vision, containing visual pigment rhodopsin, and this contains the visual pigment iodopsin. This is again the shape to show most important outer segments, which is cylindrical and tall in case of rod, while it's triangular and short in case of cone. A nucleus present at different levels in case of rod, and small and dark, while this is large, present at one level, at the level of the outer limiting membrane. Again, now what's the outer limiting membrane? You can see this line, outer limiting membrane. It's formed of what? It's formed of junction complexes between Muller cells and rods and cones. You can see here a nice diagram to show you these blue cells are rods and cones. Yes. This cell, yellow in color, extending from the outer limiting membrane to the inner limiting membrane, is the Muller cells. What about these cells which will form the optic nerve fibers? Ganglion cells. What are these bipolar cells? You see, it's obvious to have two fibers, nerve fibers, dendrites and axon. These are the bipolar cell. There is an associating cells, one sharing in the outer plexiform called horizontal, other sharing in the inner plexiform called the amacrine cell. Then we'll go to layer number 3. We'll repeat again. It's not a true membrane. It's formed by junction complexes between photoreceptors and the Muller cell. These are the Muller cells, and these are the rods and cones. What about layer number 4? The outer nuclear. Now we know that when we see in histological section a band, a layer with multiple nuclei, this is the outer nuclear layer. It contains the nuclear region or the cell bodies of rods and cones. These cell bodies contain the nuclei of photoreceptors and some organelles, because cell bodies around it contain some organelles. Arrangement of nuclei, we mentioned it as regards nuclei of rods and nuclei of cones. So this is number 4, outer nuclear layer. Then what do you expect? Outer what? Outer plexiform layer. Outer plexiform layer, or layer number 5, it's synapses between. Which part? Axon terminal, synaptic contact terminal of rods and cones, together with the dendrites of bipolar, the above layer above, bipolar cell, and horizontal cell, sharing with the processes of horizontal cells. So the plexiform layer is synaptic contact between the axon terminals of rods and cones and the dendrites of the bipolar cells with the horizontal cell. If we look at this diagram, you can see the rods and cones here and nuclear layer here. Then this is outer nuclear, this is outer plexiform. Synaptic contact between what? Axon terminal of rods and cones together with dendrites of bipolar cell, together with a cell called horizontal cell. Then what do you expect? What's the next layer? Layer number 6 is, we finished outer, now we start with inner. Inner nuclear. Inner nuclear, remember, 4 cells. Let's now discuss the histology of the bipolar. These are the bipolar cells. You know, cell body, then one fiber will act as a dendrite, the other will act as an axon. So it extends from the outer plexiform to the inner plexiform. The dendrites contact with the synaptic ending of rods and cones in the outer plexiform. Look at synaptic contact of rods and cones together with the dendrites of the bipolar and the processes of the horizontal cell. While, this is about the dendrites of bipolar, while the cell bodies and nuclei are present in the inner nuclear layer, while the axons of these bipolar cells will synapse with the dendrites of the ganglion nerve cells in the inner plexiform layer. So it extends from the outer plexiform to the inner plexiform, and cell nuclei are present in the inner nuclear layer. Remember, outer nuclear layer contains the nuclei of rods and cones. What's this cell which extends from the outer limiting membrane to the inner limiting membrane? Yes, these are the Muller cells, the supporting cells. Now, number 6. Here, this is the outer nuclear layer. This is the inner nuclear layer, in between outer plexiform layer. This is the inner nuclear. 4 types of cells. Remember, inner nuclear layer encloses 4 types of cells. The most important is bipolar. Yes. 2 associating cells, what? Horizontal cells and amacrine cells. And then the supporting cell body lies here. This is the Muller cell. 4 cell bodies. Then we finished the inner nuclear layer. We'll go to the inner plexiform layer. Inner plexiform layer, it is synaptic contact between axons of bipolar and dendrites of the ganglion cell layer. You see, these are large ganglion cell layers. There is interplay between, or synaptic contact between, the ganglion, dendrites of ganglion, and axons of the bipolar together with the amacrine cell. So amacrine cells and the bipolar cells together with the dendrites of the ganglion cell. Number 8, this is the ganglion cells. You can see large size cells with more or less vesicular nucleus. Okay, they occupying this layer, ganglion cell layers. Their dendrites share in the synaptic contact in the inner plexiform layer, where their axons come here perpendicular and then bend to form the optic nerve fibers, which will pierce the sclera at the lamina cribrosa and go out as optic nerve fibers. And as I said, it will be myelinated without neurolemma by the oligodendrocytes. So these are layer number 8. Then we'll go to layer number 9, which is the optic nerve layer. What are these optic nerve layers? Look at this. This is the ganglion cells, and these are the nerve fibers, which optic nerve fibers, which are the axons of the ganglion cell. The fibers appear pale in histological section. This is layer number 9. Then limited the retina internally by the inner limiting membrane. What's the inner limiting membrane? It limits the retina from inside, separating it from the vitreous. And it's made up of what? Basal lamina of Muller cell. That's why we said that the Muller cell extends from the outer limiting membrane to the inner limiting membrane. These are the cells of the retina. These are the layers of the retina. The green one, rods and cones. The orange, bipolar. Yellow, one is horizontal. Maybe this one is green or yellow to green. This is the amacrine cells. These are the ganglion cell. And what about the violet one? These are the Muller cells. Now we finished histology of the retina. Accessory structures of the eye. Other than the three coats of the eyeball, other than the lens inside, we have accessory structures, including a conjunctiva, which is a thin, transparent mucous membrane, as I said, made up of non-keratinized stratified squamous epithelium with few goblet cells. There is a variation in the lining epithelium of the conjunctiva. Now we know that the conjunctiva is covering the anterior portion of the eye up to the retina. So this is the conjunctiva never to cover what? The retina. The cornea, sorry. The cornea is not covered with Now we finished the three coats of the eye and the lens, and we'll start accessory structures of the eye. Accessory structures of the eye include the conjunctiva. We mentioned the conjunctiva as a thin, transparent mucous membrane. And this mucous membrane covers the anterior portion of the eye up to the cornea. You have to know that the cornea is not covered by conjunctiva. Only the sclera, the white of the eye, is covered with the conjunctiva, and this part of the conjunctiva covering the white of the eye, the anterior portion of the eye up to the cornea, not covering the cornea, is called bulbar conjunctiva. But the rest of the conjunctiva will line the two folds, the upper eyelid with lower eyelid, and this part of the conjunctiva will be called palpebral conjunctiva. What's about the lining epithelium of the conjunctiva? It varies from non-keratinized stratified squamous epithelium to stratified columnar epithelium with goblet cells, mainly in the bulbar conjunctiva and in the junction between the cornea and the sclera. It's covered with non-keratinized stratified squamous epithelium. At the margin around the cornea, non-keratinized stratified squamous epithelium, also the border of the eyelids, non-keratinized stratified squamous epithelium. But in the palpebral conjunctiva, especially at the fornix, the epithelium will be stratified columnar epithelium with goblet cells. You can see here, that's what I mean. This is the bulbar conjunctiva, stops at the cornea. The conjunctiva doesn't cover the cornea, stops here. So this is the bulbar conjunctiva, and here the lining epithelium of non-keratinized stratified squamous epithelium. While the part of the conjunctiva which lines the upper and lower eyelid from inside is called palpebral conjunctiva. The fornix is here, the junction here, and this is formed of the stratified columnar epithelium with goblet cells. So the conjunctiva is reflected to cover the internal surfaces of the eyelids. We call it palpebral conjunctiva. Conjunctiva main function is to lubricate and defends the eye against infection. Okay. Now we'll discuss the histology of the eyelids, and in the eyelid we have four main layers. We have a skin surface. You know, we have an upper eyelid and lower eyelid. From outside, it's covered with skin surface, and we know the histological structure of skin. Its epidermis is keratinized stratified squamous epithelium with hair follicles. And then we'll go to inside, we find muscles. We have two types of muscles. We have skeletal muscles and we have smooth muscle fibers. The skeletal muscle fibers are orbicularis oculi muscles, which closes the eye, and the levator palpebrae superioris, which elevate the eye, the eyelid. while we have the superior tarsal muscles or molar cells, which are smooth muscles, and it's supplied by sympathetic nerve. This is embedded in what's called, in the superior part of what's called tarsal plate. What's this tarsal plate? Tarsal plate is a layer of fibroelastic layer formed of collagen fibers and elastic fibers. And embedded in this tarsal plate gland called meibomian glands or tarsal glands. This meibomian glands or tarsal gland are modified sebaceous gland. And so you know the sebaceous gland secretes an oily secretion. What's the importance of this oily secretion? We find that this oily secretion will be added to the surface of the eye together with tears. And so they form, these tears can resist evaporation when it's mixed with tears. It will prevent rapid evaporation of tears. Then from inside, the most inner layer will be the palpebral conjunctiva. As we said, the palpebral conjunctiva is discuss the histology of the eyelids. No upper eyelid and lower eyelid. What are the layers of the eyelids or histological structure? From outside, it's covered with skin. And we know skin, epidermis of the skin, keratinized, stratified squamous epithelium with hair follicles. And then we'll go inside, we'll find muscles. There are two types of muscles. There are skeletal muscles and smooth muscle fibers. Skeletal muscles include the orbicularis oculi muscle, which closes the eyelids, while the levator palpebri superioris, and this levator palpebri superioris elevates, this is for elevation, and while we have smooth muscle fibers, which is superior tarsal muscles, or we call it Muller's muscles, which is innervated by sympathetic nerve and inserted in the tarsal plate. What's the tarsal plate? Tarsal plate, as you see here, it is formed of fibroelastic tissue. This plate is formed of fibroelastic tissue in which embedded group of glands. We call them meibomian glands or tarsal glands. What's the function of these glands? They are modified sebaceous glands. Modified sebaceous gland, you remember, these glands secretes oily secretion, and this oily secretion will be added to the tears on the front of the eye, and so this oily secretion will prevent the rapid evaporation of tears, okay? Help to keep the tears in front of the eye by their oily secretion. Then from inside, the eyelids lined with palpebral conjunctiva, as we said, palpebral conjunctiva. This is the part which lines the inner part of the eyelid. Here another diagram to show you the skin surface, the palpebral surface, the muscle fibers, and the tarsal plate, and the meibomian glands tarsal glands are present here, and we can see the red margin and protruding from the red Here this is a diagram for the eyelids. We can see here the outer surface, this is the skin surface, the inner surface, this is the palpebral conjunctiva, and the muscle fibers are present here. The tarsal plate showing the meibomian glands or the tarsal glands, which are modified sebaceous gland, and the lid margin, you can see from the lid margin protruding the eyelashes. Then what about the glands of the eyelids? We said meibomian glands in the eyelid, we discuss it. Now we'll discuss the lacrimal gland. What's the lacrimal gland? This is a tear secreting gland located in the anterior superior temporal portion of the orbit, and it's conducted by its duct to the superior conjunctival fornix. Okay, so these glands will secrete the tears, and the type of the glands is serous glands. The secretion causes moistening of the corneal surface and the conjunctiva. Tears also has a bactericidal effect, rich in lysozyme content. We have to know the pathway of tears. The tears, as I said, here, secreted from here, from the lacrimal gland, and open into the fornix of the upper eyelid. Then it will be drained into, first, lacrimal canaliculi. These are the lacrimal canaliculi through two puncta here. So the first is lacrimal canaliculi. This is the pathway of drainage into lacrimal sac. This is the lacrimal sac. Then nasolacrimal duct to open into the inferior meatus of the nose. So from lacrimal canaliculi, nasolacrimal duct, or the lacrimal gland, its secretion causes moistening of the corneal surface and the conjunctiva. Tears have bactericidal effect, rich in lysozymes content, and the secretion is drained here by lacrimal canaliculi. Here are the tears secreted from the lacrimal gland. It will go into the fornix of the eyelid, will go to the surface of the eye, and will be drained by, first, lacrimal canaliculi through two puncta here. These are the lacrimal canaliculi into lacrimal sac, and then through the nasolacrimal duct to open into the inferior meatus of the nose. This is the drainage of the tears, and this will end our lecture about the accessory structures of the eye. Thank you so much.